Good morning, everyone, and welcome. Today, we're diving into a comprehensive overview of gene expression and microarray analysis. We'll explore the key techniques used in this field, discuss their important applications, examine some of the challenges researchers face, and take a look at future perspectives in molecular biology. I'm excited to share this fascinating topic with you and explore how these tools are shaping our understanding of genetics and biology. Gene expression analysis is all about examining how active specific genes are within cells or tissues. This gives us detailed information about how genes function and are regulated. By measuring the activity levels of these genes, scientists can gain a clearer understanding of the biological processes happening inside cells, as well as the mechanisms behind various diseases. This method is crucial for studying how cells work, how organisms develop, and how cells respond to changes in their environment or to different treatments. It provides a powerful window into the inner workings of life at the molecular level. When analyzing gene expression, there are several key methods we can use. First, quantitative PCR or qPCR is a targeted and highly sensitive technique that allows us to measure specific gene transcripts with great precision. Next, RNA sequencing or RNA-seq provides a comprehensive profile of all RNA transcripts in a sample, giving us broad and detailed information about gene expression patterns. Then we have northern blotting, a traditional method that detects specific RNA molecules, useful for verifying expression, but less commonly used now due to more advanced technologies. Finally, DNA microarrays enable the simultaneous measurement of thousands of genes by using chip technology, making it possible to study large-scale gene expression changes efficiently. Each method has its strengths depending on the research question and the level of detail required. DNA microarray assays work on the hybridization principle, where labeled complementary DNA or RNA molecules bind specifically to matching DNA sequences that are fixed on the surface of a solid chip. Each spot on this chip represents a distinct gene sequence, which enables us to analyze thousands of genes at the same time. The key thing to understand here is that the intensity of the signal we detect from each spot directly reflects how much of that particular gene's transcript is present in the sample. So, essentially, this method allows us to capture a simultaneous snapshot of gene expression across a vast number of genes, giving us a powerful tool for broad and detailed genetic analysis. First, we start with sample preparation, where RNA is isolated from the cells or tissues we want to study. This RNA will serve as the basis for our analysis. Next, we move to the labeling step, where fluorescent dyes are incorporated into the nucleic acids. These dyes are crucial because they allow us to detect the presence of specific sequences later on. After labeling, we proceed to hybridization. Here, the labeled RNA is applied to the microarray chip, which contains many probes. These are short DNA sequences designed to bind to their complementary targets. If the RNA matches a probe, it will bind to it. Once hybridization is complete, the chip goes through a washing step to remove any unbound or loosely bound material. This is important to minimize background noise and ensure that our detection is as accurate as possible. Finally, we scan the chip to detect the fluorescent signals at each spot. The intensity of fluorescence at a particular spot corresponds to the level of expression of the gene represented by that probe, giving us a snapshot of gene activity in the sample. When interpreting microarray data, the first crucial step is normalization. This process adjusts for any technical variations that might occur during the experiment, ensuring that we can accurately compare results across different samples. Next, we focus on signal detection, which involves distinguishing true hybridization signals from background noise. So we only analyze meaningful data. After that, differential expression analysis helps us identify which genes show significant changes in expression under different conditions, highlighting potential areas of biological interest. Finally, statistical and bioinformatic analyses are essential to make sense of the raw intensity data, allowing us to extract valuable biological insights and draw reliable conclusions from the experiment. Gene expression and microarray analysis have a wide range of important applications. One key area is disease profiling, where we use these techniques to characterize molecular signatures in conditions like cancer and neurological disorders, helping us understand the specific changes at the genetic level. In drug discovery, Microarrays allow us to identify which genes are affected by new therapeutics, guiding the development of more effective treatments. Another crucial application is biomarker identification. By detecting unique gene patterns, we can predict disease progression or how a patient might respond to a particular treatment. These tools also play a big role in genetic research, deepening our knowledge of gene regulation and the complex networks that control cellular function. Finally, microarray analysis helps us study cellular responses, showing how gene activity changes in response to environmental shifts or medical interventions, which is vital for both research and clinical applications. When working with expression data, one major challenge is the variability that comes from both biological differences and technical factors. This variability can make it harder to draw clear conclusions. Another issue is cross-hybridization artifacts, which can lead to false positives and lower the specificity of our results. Additionally, the limited dynamic range of the technology means we might miss very low or very high expression levels, restricting our ability to detect the full spectrum of gene activity. Technical sensitivity also plays a role. It requires us to maintain strict quality control throughout the process to ensure reliable data. Finally, interpreting this complex data isn't straightforward. It demands advanced computational tools and specialized expertise. Being aware of these limitations is essential for designing experiments thoughtfully and analyzing results accurately. As we wrap up, it's important to remember that gene expression and DNA microarray assays continue to be fundamental tools in molecular biology, giving us valuable insights into how genes function and are regulated. Technology and analysis methods are always evolving, making these assays more accurate and allowing them to be combined with other omics data to broaden their applications. Looking ahead, efforts are concentrated on boosting sensitivity and overcoming current limitations to make these tools even more effective. One of the most exciting directions is applying these advances in personalized medicine, where diagnostics and treatments can be customized based on an individual's unique gene expression profile, bringing us closer to truly tailored healthcare solutions.